Thank you very much for the invitation to speak at this important meeting. I was here at the last meeting, and it was very inspirational for me to be able to meet and talk to the patients and families who are struggling with this disease that our WELLSTONE Center is dedicated to understanding and developing therapeutics for. As you know, the WELLSTONE Centers, there are 6 national centers, and there's one of them dedicated to FSHD research. That's our center. And I'd just like to tell you about 4 accomplishments that we think are significant that we've achieved at the center in the past several years that I think you'll find of interest and provide further evidence to what we've heard in the first 2 speakers about the momentum that's building in this area of disease research that's going to lead to development of therapeutics in the near future. So our center, as all WELLSTONE centers, are multidisciplinary and collaborative. And you all know as patients with this disease how complex it is and how different you are from another patient. It's a very complex disease. So to attack this disease, we believe we have to assemble a team that is multidisciplinary and collaborative. No one person can really attack this kind of complexity. And so the WELLSTONE Centers and our center brings together patients, scientists, clinicians, and companies, and the goal is to develop therapeutics for FSHD. So our team is a collaborative team, and it's multidisciplinary. I'm not going to go through everybody on the team, but myself, Lou Kunkel, are the directors, and Catherine Wagner, whom many of you are familiar with, is an outstanding neurologist and clinical scientist, and really the person that deals with a lot of you as a patient, an excellent physician, and you should know is a terrific scientist as well. And then we have other members of our team who bring to us significant expertise in areas that we believe will be important in solving this disease. In addition, we have Dan Perez in the FSH Society, the sponsor of this meeting, who is integrally involved in the leadership of the center, as well as several companies that are involved in working with us to develop ideas for therapies for the disease. So the goals of the center are I'd just like to give you, because that's the reference for the accomplishments that we've made. One goal is to create a muscle cell biopsy and cell repository from patients, and many of you in this room have participated in this study, to get the statistical power we need to really study this disease. Very important. This has not been done before, and the center's one of its primary missions is to gather enough biomaterials to be able to do critical research in the field. Second goal is biomarker discovery. We're interested in understanding what the disease gene is. It DUX4? Is it some other gene? You've heard a lot about DUX4, and we'll talk some more about that in a minute. And then how do we develop biomarkers we can use in therapeutics? That is, to develop new therapeutics for the disease, ways to monitor drugs as well in preclinical studies, as well as ways to monitor the effect of drugs in clinical trials, which many of you will be participating in when the time comes. Then the third goal is to develop models for FSHD that will allow us to develop drugs, cell and animal models. And we'll talk a little bit about that too, and what we're doing at our center to achieve that. As you know, FSH is a very complex genetic disease that involves a locus which is not present in mice, and so the traditional approach to developing a disease model through mouse models is very complicated. And so we're trying to develop some really innovative ways to think about how to do that. And then finally, our center is to be a collaborative hub to promote collaborations amongst scientists, and you'll see some examples of that, how we're trying to help bring everybody together and towards breakthroughs in therapeutic development. And so we've hosted some meetings, particularly the International FSH Workshop, and we're also sponsors of this meeting here today. So let's talk first about the biopsy and cell repository program. We have, at the beginning, a very specific strategy to collect biomaterials from patients and family members with FSHD. We call this the family cohort approach. where we've asked families to participate by donating samples of deltoid muscle and bicep muscle tissue for us to analyze. And so we've asked families of members who are not affected by disease and those who are affected by disease to make these donations. And then with this material, we do an array of studies, including making cell culture models of stem cells and satellite cells from the muscle tissue that we've recovered, as well as doing gene expression, genotyping, proteomics, and a variety of studies to really characterize the pathology of the disease. So we choose deltoid muscle because often, although not always, it's less affected in patients in the initial stages, and biceps, which tends to be much more affected. So we are able from any one patient to study a muscle which is more diseased than another muscle within the body, as well as family members who do not have the disease but are genetically related as first-degree relatives to the patients, which is very important in understanding disease because we are all different as human beings. And so families are more similar to one another genetically than not. And so many of the differences we see, we want to be able to that are maybe related to disease, we want to be able to subtract out from that's related to genetics of family. So, wow, what's happened? We've worked with 36 families. I think by now I show 34 here who've provided a family member who has had the disease and has the disease to give muscle tissue, as well as a family member who is not showing disease. This is amazing. So we've collected both bicep and deltoid tissue, and Catherine Wagner, as we'll see in a minute, the team that does this has done the collection of this material, and as many of you been the physician that's been treating you for the disease. So this is a remarkable achievement because this now allows us to do significant, statistically significant research in the field with these biomaterials. So what are the accomplishments? We've now collected 28 families, complete cohorts, that is enough to give us enough power for biomarker studies. And interestingly, 5 families have come to us for donation of materials, and one of the family members that was a control in the study, showing no disease effect, turns out to have FSHD1 deletion of their genome. In other words, is actually genetically an FSH patient, but shows no disease progression, even some that are 68 years old. So this is very interesting, and I'll come back to this point later. And it again shows you the complexity of this disease, that the DNA genetics and the pathology of the disease are not coordinate with each other in all cases. We've made cell lines, stem cell cultures from these materials that are very important, cell models for studying this disease and getting an analysis of candidate biomarkers such as DUX4, which I'll talk about in a minute. We've made immortalized cells from these, from 4 of these cohorts. That is, we've been able to engineer these cells so they will grow indefinitely in culture, a very important tool for screening drugs to be able to have a cell model in which is not changing and will be immortal to long-term drug screening, such as you just heard about in the second talk. And then finally, we've distributed 7 of these family cohorts to 9 laboratories worldwide, and we'll distribute them to any laboratory that asks. So we have made biomaterials that are available to the field as a whole for doing studies in which there's sufficient statistical power to get meaningful results. So here's the team that did it. And we've got, of course, Catherine's lab at Kennedy Krieger, and my group at Boston, and Woody Wright at UT Southwestern, who does the immortalization. But most important is the FSH Society, who knows you all and can identify families who can participate in the study, and you, who've actually been willing to participate in developing this really unique resource. So thank you so much for your efforts, which are going to be really key in making progress in this field and already have been. So what are the goals of the center? Let's talk about So that's one accomplishment, is making this repository and having cell models for this disease and biopsy material for analysis. The second, I want to talk about three sort of accomplishments that we've made using this unique resource in our group, the WELLSTONE Center, to give you a flavor of what the kind of questions that we can ask uniquely with this unique bioresource. So we've been studying families that have FSHD1, and that's shown here, this second here. We're studying families with deletions of the D4Z4 repeat between 2 and 7 copies left, and with the A allele, which gives you the polyadenylation site that we've heard about that's important for the disease. And also we have family members who have B alleles, which are important controls for our work. So that's what we've been focusing on in our repository building and our studies. Now, the first thing I want to talk about is, with using this repository, is asking the question, is DUX4 a sufficient cause of FSHD? In other words, we've heard from the first two speakers about this molecule, this protein that's expressed from the terminal repeat that's left in a deletion of the D4Z4 repeat. We've heard about the important evidence that that gene is in fact an important player. But with our repository, we're able to ask whether that gene is actually correlated and sufficiently insufficient and always present in patients with FSHD that show pathological symptoms. So, as we heard from Dr. Tapscott, there are ways to assay DUX4 transcripts, RNA, and we're particularly interested in this important work that Stephen talked about from his group and his collaborators on this FSH long form, which is seen on this analytical gel that shows you we can assay the RNA for DUX4 long, which is the toxic transcript that we've heard about that causes cell death. And we also can assay DUX4 protein using antibodies. And again, this shows the power of collaboration that Stephen Tapscott's group has made to DUX4 that is shared with us in our WELLSTONE Center to be able to characterize DUX4 expression in our patient population. And so we've done a study of 19 families looking at expression of DUX4 protein in RNA in cells and in biopsy material. And I want to spend some few minutes telling you about these results. So the question is whether DUX4 is expressed uniquely in FSH muscles that are undergoing pathology. So we looked at, in these 19 families, we looked at 4 classes of patients. One, symptomatic patients that show muscle disease. Asymptomatic patients, that's at 5 families where there are members who have the deletion but don't have any signs of pathology. And then we have unaffected controls that have normal D4Z4 locus, no deletion in the family. And then this individuals that have the B locus, which means they have no polyadenylation, so there's no way to make a DUX4 protein. So they're nice negative controls. So on the left axis, this is just measures some amount of protein that's being made of DUX4. And on the bottom axis, we looked at 4 different tissues from these patients, these 19 patients. ABIC means affected patient with bicep. UBIC means unaffected muscle, a patient with in the bicep. And ADEL means affected deltoid muscle, and U means unaffected deltoid. So if we look at the red dots show symptomatic FSHD patients. These are patients among you in our repository who have muscle disease. And you can see that by and large in the ABIC, just look at that first column, you can see that the red dots, mostly show pretty high level expression of DUX4. And so that shows good correlation between that in bicep muscle as well as ADEL, which is the deltoid muscle. But there are exceptions. There are individuals that in the ABIC, there's one down there that shows no DUX4 expression at the very bottom, that's 0, and ADEL, which shows, again, the red triangles, shows no expression of DUX4. So there are some symptomatic FSHD patients in our large collection that don't show any, in our assays and biopsies in cells, any DUX4 expression. All right, so that's the first sort of interesting observation about an imperfect correlation between DUX4 expression and FSH pathology. Second is, if you look at the green dots in ABIC, you can see that there are asymptomatic individuals who have the deletion but don't show any symptoms of disease pathology, which express lots of DUX4. So DUX4 expression in those individuals is not leading to any pathology. And then third thing is, if you look at the U columns, you can see that people that don't have a deletion of the D4Z4 locus, these are control members of families, some of them can express some levels of DUX4 already, and those are the black dots you see in UBIC and UDEL. So, and then you can see with the final one, the 4QB, that the blue dots, that none of them can't make a transcript, and none of them show DUX4 as a really negative control. So what these data show are that asymptomatic patients, that expression of DUX4 is not perfectly correlated with disease. And this is we think, a very important observation. This does not challenge the idea that DUX4 is an important player. But what it suggests is that there's protective mechanisms that might keep DUX4 under control, suppressors of the process. Mother Nature may have created a therapy for this muscular dystrophy already by having genes that are present, the suppressor genes that keep the toxicity of DUX4 under control. And this kind of thing is seen in cancer. You've heard about oncogenes that cause cancer. Then there are tumor suppressor genes that suppress cancer formation. So this suggests that there are mechanisms that may ameliorate the effects and protect the effects of against DUX4 in the genome of individuals, which is very important, because if we knew what those protective mechanisms are, we would have a therapy. So understanding the mechanisms that lead to protection against DUX4 toxicity and the genes that might be involved is a really important endeavor that can now be undertaken with the genome techniques that are available. So the second accomplishment in the biomarker discovery area is that we've done a genome-wide microarray analysis, that is looking at all the transcripts that are produced from genes in FSHD cells and control cells in our family cohorts. And this has led to the identification of a large set of disease genes, which turn out to be extremely interesting, and in the sense that they bring together the work from the Tapscott group and that group that he's working with on DUX4 target genes and disease genes that can be identified by this more undirected screen that we've done, looking for disease genes in our cohorts. And I'm going to try to take you through these data, because I think they're quite interesting. And Stephen has not seen this yet, but he's heard about it. So here we have some plots. On the bottom plot is a plot of gene expression. Each dot is an RNA transcript, a level of expression in a DUX4 overexpressed cell. He's taken a virus and taken a muscle cell and put in a lot of DUX4 and then looked for the expression of individual genes. So as you go to the right from the center of this configuration here we go, here's the center. As you go to the right, these are genes that get really activated when you overexpress DUX4. And so there's a whole bunch of them, and he talked about them. They're involved in germline development. Now on this, so that's on this axis. What genes get activated when you really put a lot of extra, artificially put DUX4 in? There's a whole bunch of genes. On this axis is what we found when we did our totally independently our array analysis of of genes that are expressed specifically in FSHD cells and not in control cells in our collection of symptomatic patients. And what we can see is that, look at these genes that get upregulated. So anything that's up in the upside, these are overexpressed in disease cells, and they turn out this box on the right-hand side that's in red is the genes that Tapscott identified in his screen. So we know in FSHD cells, normal from patients, we can get the same genes, DUX4 overexpressed genes, a large set of them, about half of them, that are DUX4 target genes that he identified in this overexpression artificial screen. This brings together one of the first examples of where we're bringing together data from two different groups that comes together and shows that we really agree on something. And this has been a problem for the field, and part of this is driven by the fact that we have enough material now to really ask these statistical powered questions. So we have a whole bunch of genes, but then look, we also identified, if you look in the middle, the up and down, the blue boxes show us that we've identified a whole bunch of genes that go up and genes that go down that aren't DUX4 target genes, because they're not to the right or the left of the center line, they're up or down. So we have an additional 50% of the genes that we've come up with that are FSH correlated. These could be genes that have to do with modifying disease progression. These could be genes that are involved in why muscle cells die, but they're not DUX4 target genes. So in addition to the DUX4 target genes, we've come up with another collection of genes that are clearly correlated with FSH disease. And so we have now two collections, DUX4 targets and then up and down regulated other genes that aren't in the DUX4 family that are associated with this disease. This is very important advance, we believe, because it provides the tools that we need now to ask very specific questions about not only DUX4 function in disease, but also the function of these other genes in the pathology of the disease. We can ask questions like, what's the difference between unaffected and affected muscle in terms of gene expression? Do all these genes go up and down if you have a deltoid muscle versus a bicep? What about manifesting versus non-manifesting individuals that have the deletion that make DUX4 but don't have any disease? Do they express the DUX4 target genes and these other genes? And then finally, what are the new tools that we can use these as tools to monitor therapeutics in clinical trials? If you use a drug, you should be knocking these disease genes down or restoring their normal function. So this is a very important advance, we believe, in the field that brings together the work on DUX4 with a broader spectrum of genes that we can study involved in disease and therapeutic development. The final advance I'd like to talk about is some work that Catherine Wagner's group and my group have been doing to develop a unique animal model for therapeutic testing of FSHD. And this model is based on our repository of being able to develop biomaterials that allow us to isolate stem cells from muscle and from FSH muscle, as well as in our cell repository, as well as isolating muscle fibers. I'm going to talk about our work, which is a cell-based work, where within the general idea is, can we take FSH cells from and their family member control cells and engraft them into mice and replace mice muscle with FSH muscle. So we have a mouse model, a humanized mouse model for studying this disease. And so this technology is out there for engraftment of cells into mice using immune-compromised mice. So the idea is to take our FSH and control cells from our repository and engraft them into mice in which the muscle, the TA muscle, is on your back of your leg there, is damaged and has to undergo regeneration, and then rebuild it with human muscle stem cells. And I want to show you that this is working in ways that we think will be very important to test the regenerative potential of FSH cells, as well as the pathology of SSH fibers that form in mice. And so this makes no assumptions about whether DUX4, this technology, about whether DUX4 is the right gene for a mouse model or not. We're putting the human FSH cells into mice and letting them form human muscle in mice. So this is quite interesting. And here's an imaging study of a whole mouse, and you can see from this on the left mouse, you can see this spot here where we put a luciferase reporter, a visual reporter in these cells and graft them into the muscle, and by golly, they actually engraft and start rebuilding human muscle in the mouse. And you can look in the tissues of these muscles with human markers and show that the human cells are actually rebuilding muscle fibers and actually have myogenic have stem cells in them too, PAX7 positive cells. So in any case, this technology is really going to, we think, open the door to do therapeutic testing on real FSH muscle in an animal, as well as to understand the pathology of the disease. And so I think that's going to be an important advance. So we're meeting our goals. We've developed a repository for the center. We've used this repository to do biomarker discovery around DUX4. It gives us a chance to test that theory in much more detail than previously and develop new biomarkers for FSH, and also to develop animal models for the disease. So all of this work is been supported by FSH Society and NIAMS, NIH, and Child Health and Muscular Dystrophy Association. But the most important thing is it's been supported by you, the families and patients, because you've given us the materials that really are allowing us to break open this field. So thank you very much.